Welcome back to MLT 240. Today we're going to cover the last lecture for this PowerPoint on sporozoans. In general, some characteristics is they're obligate endoparasites, meaning that they have to have a host to complete their life cycle. Endo meaning they're going to live inside their host. They do not have any apparent organelles for locomotion. Most produce an infective spore that is ingested by the host, or it's injected by a biting arthropod vector. In general, they have a life cycle that includes sexual and asexual phases of reproduction, and most have a two-host life cycle. Here's a list of the sporozoans or parasites that we're going to cover in this unit. The first two are blood sporozoans. First up is Plasmodia. Plasmodia is the one that's probably the most important and the one we're going to talk the most about. This is the parasite that causes malaria. Remember back at the beginning of this PowerPoint, we said that about 500 million people end up with malaria each year. Next for blood is Babesia. For the tissue sporozoans, we're going to talk about Toxoplasma gondii. For the lungs, or sporozoa that affect the lungs, we're going to talk about Pneumocystis carni. For intestinal protozoans, we're going to talk about Cryptosporidium parvum and Cyclospora bellii and Sarcocystis and Cyclosporia. So let's start out with Plasmodium species. There's a few different species specific we're going to talk about here in a minute, but let's just talk in general. There's stages that are going to develop within the peripheral blood of the human host. First, you're going to have the troph or trophozoite stage. You can often see ringlets or rings in the red blood cells of the trophs. You can see in this image that the troph stage takes over most or occupies most of the volume of the red blood cell, so it kind of hijacks your red blood cells. You can see there's a presence of a pigment that is in the parasite. So this is a growing trophozoite, but you're going to end up with a smaller ring structure we're going to look at in just a second. Here's a better image of what the ring structure looks like of the mature trophozoite stage. Another stage of development for the Plasmodium species is the schiznot stage. This is a multinucleated form of the parasite, and this is where asexual reproduction happens, and it happens through nuclear fission. So let's look at an example of what it looks like. This would be what the schiznot stage looks like. You have several multinucleated forms within the parasite. Something to note before we leave this image, we're going to discuss what a merozoite is next. So this little red dot inside each one of the forms of the parasite is the merozoite. So the merozoite is the nuclei in the mature schiznot just prior to the rupture of the red blood cell. So the red blood cell is going to rupture and release all those forms of the parasite. The last is the sexual phase. This is the gametocyte. It is infective and it transfers, and is the transfer stage to the female mosquito. And within our different species of Plasmodium, the gametocytes are shaped a little differently. Within Plasmodium vivax and Plasmodium malariae, the gametocyte is oval to roundish in shape. But with Plasmodium falciparum, it is crescent shaped. So I know I just threw a lot of information at you about the life cycle or the different stages of development of Plasmodium, but let's look at the general life cycle on this diagram. So let's start out here at number 1. where the mosquito takes a blood meal from our human host. Okay, so there's our little mosquito. It bites the human host. Well, the first place that it goes is the liver cells of the human. We don't go directly to the blood. We're going to go to the liver cells, and it's going to affect the liver cells. And the schiznot form is going to form first. Okay, so this is known as the exoerythrocytic cycle. So we're not in the blood yet in this particular phase of development. So our schiznots have an option here of going ahead and rupturing, or we can go on down and start affecting the red blood cells. So here's our little red blood cell. The parasite goes on and we go into the troph stage, and this is the ring stage. This is where we can start to actually see rings within our red blood cells for the trophs. But in the blood you can also see the schiznoid stage, and remember this is asexual reproduction here by binary fission. And remember, the merozoites are those nuclei within the schiznoid. So you can also have rupture here and release of the parasite. So you just continue with this cycle until they decide they're going to start their gametocyte reproduction or sexual reproduction phase. And remember, the different species of Plasmodium have different shapes for their gametocytes or their sexual phase. The falciparum has this little crescent shape, and the others have more of an oval or round shape to their gametocytes. Once that sexual reproductive phase happens, that's when another mosquito can come along, bite this same person, and that's when they're going to pick up those gametocytes. And that's when the process can start again in this mosquito for all the stages that happen within the mosquito. So that mosquito can bite somebody else, and the whole life cycle can start again. So here are some of the different species of Plasmodium that we're going to talk about and look at. There's vivax, there's falciparum, there's malariae, there's ovaliae, and then there's novicii. And again, all these cause malaria. The mode of transmission is a vector or a mosquito bites the host. And the species of the mosquito is Anopheles. So it's through blood transfusion that is the way that people get malaria or Plasmodium. The distribution is usually tropical, and if people in the U.S. end up with it, it's usually because they've been somewhere and it's being imported in. As far as the species distribution, vivax is the widest distribution. Ovaliae is confined to Africa, but the others are found in Africa and tropics or tropical areas. For laboratory diagnosis, we collect a EDTA whole blood sample, and we want to do slides within 1 hour of collection. And you usually want to collect between paroxysm and febrile episodes, meaning disease and fever episodes. For your smears, you're going to prepare one thin smear and one thick smear. The thin smear is more for scanning, and you're going to prepare it for a differential. You're going to view 100 fields for 30 minutes. The thick smear is for differential diagnosis, and this is the gold standard for malaria. You want a clean, unscratched slide, and you want it to have been protected from dust. As far as the sample size, you want to use a blood drop the size of a dime, and you don't want it so thick that it cracks while drying. You want a thick smear, but you don't want it so thick that it's going to crack. As far as the stain that's used or recommended for malaria, you want to use the Gansen stain. but a right stain is acceptable. And it must be repeated, the testing must be repeated every 6 to 12 hours for at least 2 to 3 days. Signs and symptoms and pathology for malaria. It's the major cause of mortality in underdeveloped countries. Remember in the beginning we said of that almost 500 million cases, about 1.5 end up dying from malaria. It causes fever and chills, especially during those times when the red blood cells rupture and release those Marley-Trotter because it causes toxins or toxin release in the body. Initial symptoms can be anywhere from headache, fever, nausea, even pain in your bones. It can cause shaking and chills every 10 to 15 minutes. There can be a spike of a fever up to 104 to 106 degrees Fahrenheit. This can last for 2 to 24 hours. There can be a cycle that repeats at regular intervals depending on the species of plasmodia that you're infected by. You can have damage to the liver and the spleen, and of course because it affects your red blood cells you can have anemia. Some of these symptoms we've already talked about, so I'm just going to hit on some of the things or point out some things that I want you to know. As far as intervals or cycles, it's usually a cycle of about 48 hours for Vivax, Valier, and Falciparum, and the cycle is about 72 hours for Malaria. And then you can also have interference with oxygen utilization. Remember, your red blood cells are carrying your oxygen. Complications with Plasmodium falciparum can lead to hemolysis and hematuria, or blood in the urine, due to a severe immune reaction. Also, agglutination of parasitized red blood cells, so they might start sticking together. Also, formation of thrombi or clots in your vital organs, like your brain, lungs, and kidneys. So, patients that have malaria tend to feel pretty rough, as you can see by these symptoms. Plasmodium falciparum ends up being the most severe because the organisms can invade our young and our old cells. Remember, our red blood cells live 120 days plus or minus 10 days. The onset of this particular species is more gradual, but the host stage lasts longer. Episodes can last 20 to 36 hours, and it can be accompanied by delirium of the patient. Complications are known as malignant malaria. It can cause pulmonary edema, meaning there's fluid in the lungs. It can cause blackwater fever, and symptoms of blackwater fever include hemolysis, hemoglobinuria, and renal failure. You can have mass lysis of the red blood cells, which causes renal insufficiency, fever, jaundice, and persistent vomiting. It can promote physiological changes of the red blood cells, causing a knobby surface or a knobby appearance, causing agglutination and lysis of those cells. Sciogony, which is asexual reproduction through binary fission, can take place in capillaries of the brain, visceral organs, and placenta. The gametocytes are crescent-shaped with Plasmodium falciparum. In falciparum, you have what's called a double dot acoli ring form, and that's what you're seeing here, this little double dot, and you're supposed to be able to see it up in the rings, the extra little double dot in the ring forms of Plasmodium falciparum. Mainly what I want to point out on this particular slide for falciparum is here are the crescent shape for the gametocytes for falciparum, and also another thing that helps us to differentiate between the different species of Plasmodium is the number of myriazoites in the different species. So it's saying here that falciparum can have anywhere from 8 to 24 24 myriazoites in red blood cells, even though they're rarely seen. So here's a really good image showing you the different stages of development of the species Plasmodium falciparum. Of course, here would be an uninfected red blood cell. And then here is just first when the red blood cell is first affected by the parasite. And then the ring forms, and so on and so on. Here's this early schiznot form. And the mature schiznot, and all those little red dots, remember those are the myriazoites. And falciparum can have up to 24, anywhere from 8 to 24. And then finally here is the sexual reproductive phase. Here's the crescent shape of the gametocyte for Plasmodium falciparum. Next, we're going to talk about Plasmodium vivax. This is also known as tertian malaria. This is the most prevalent species. Proximus can occur every third day, so disease and symptoms can happen off and on every 48 hours, but may alternate days, initially due to two populations of myriazoites present. Relapses of disease may occur because of sequestered hypnozoites in the liver, meaning they're zoites that have been laying dormant. Also, Duffy blood and G6PD deficiency are associated with resistance to Plasmodium vivax. Duffy blood you'll learn more about in blood banking, but Duffy is an antigen in the blood. Also, sickle cell disease may play an evolutionary role in the survival of malaria. These genetic variations are incompatible with parasitic survival. So not that you would want those conditions or deficiencies, but those may help you from getting this particular type of malaria. For Plasmodium vivax, you can have red blood cells that are enlarged. You may see what's called Suffner granules. These are dots found in your red blood cells that are associated with malaria. For vivax, you're going to see anywhere between 12 to 24 myriazoites in your schiznates in your red blood cells. For diagnosis of vivax, it's usually going to only invade the younger red blood cells, and you're going to see those Suffner dots. And if you look over here to the image, you can see all these granules, or these thicker little dots within the cytoplasm. Those are your Suffner granules or dots. Your red blood cells can be enlarged up to 2 times their normal size. You can see large rings. So you can see here that this ring within the red blood cell is pretty large. So large signet ring. The trophozoite can have an amoeboid appearance with brown pigmented appearance. The skidnot can contain 12 to 24 myriazoites that we already mentioned before, and of course the sexual phase of the gametocyte is going to be round in shape. So here's a good little diagram of the vivax, or the life cycle of vivax. Here's the trophozoite, here's the skidnot stage, here's the myriazoites that have been released from the red blood cell, here's the ring stage, and then here would be an early trophozoite. Okay, now let's discuss Plasmodium malariae. This one invades your older red blood cells, and it will be about the same size as the unaffected red cells. You will not see any Suffner granules or dots in the cytoplasm. The trophozoites have a band that up here and stretches across the cell. You can see the little band here. The malaria pigment is present. The skidnot resembles a daisy petal or rosette. And here's the skidnot pattern. And you'll have anywhere from 6 to 12 myriazoites, but the average is 8. The ring form will be thick with one heavy chromatin dot. You have central brown pigment. It's the third most common species in the world, but it is less severe. And you will hear people refer to it as Quartan malaria. Here's a diagram showing the different stages of development for Plasmodium malariae. Of course, here is the red blood cell with no parasite. Here is that band stage. Here's the early band stage. I think that's what we were looking at earlier on the image on the slide. But here's that band form. Here's that daisy pattern of the schiznot stage. And of course, the sexual phase of the gametocytes are going to be round in shape. The last species of Plasmodium that we're going to talk about is Ovaliae. It's least common. This one has a single ring and it takes up about 1/3 of the red blood cell. Ovaliae affects the young and immature and even reticulocytes. The most prominent feature of this parasite or this species is the Suffner granules, and you can see them over here in this image. The gametocytes of the sexual phase is very similar to that of Vivax, and as far as the schiznot stage, it is in a rosette form and it has anywhere from 6 to 14 myriazoites. This is a really good chart that puts everything that we've talked about together for the different species of Plasmodium. So you have your different species down along the left-hand side, and you have your different phases of development. You have the ring, the trophozoite, the schiznot stage, and the gamete, the sexual reproduction stage, and you've got different examples of what each stage looks like. Plus you have this nice column over here that gives you the different characteristics of the different species. The next blood sporozoan that we're going to discuss is Babesia macarota, and this particular parasite can be found in the northeastern U.S., particularly in California. It can cause a number of different symptoms in the patients. It is the same vector as is in Lyme disease. It is the deer tick, Exodus domini, and some symptoms can resemble symptoms of malaria, and some of those things are hemolytic anemia, can be associated with Babesia. You can have asynchronous fever, and chills, sweating, anorexia, and myalgia. And myalgia is associated with pain in your muscles. As far as our lab ID for Babesia, the organisms, of course, can invade your red blood cells. You will see young trophs in a ring formation, so you can see that over here in these red blood cells. Mature trophs appear more pear-shaped and may lie in pairs. Here in this red blood cell is showing more of that pear shape of the mature troph. Also, we may see Babesia and tetrads, or what's called a Maltese cross, and that's what this arrow is pointing to right here. It almost looks like it has 4 arms coming out in different directions. There may be as many as 4 to 5 ring forms seen per red blood cell in Babesia. There's usually no change in the size, color, or shape of the red blood cells with Babesia. So now we need to discuss some sporozoans that fall in the subclass of Coccidia. Some general characteristics of Coccidia. Skygogamy, the asexual reproduction, occurs in nucleated cells of our birds and mammals. Sporogamy, or the process of making spores or spore formation in parasitic sporozoans, occur in the intestines of the definitive host. The infective form of Coccidia, the oocysts, shed in feces usually. These are primarily parasites of lower animals, so non-human, so our mammals or vertebrates. So the first sporozoan in the subclass Coccidia that we're going to talk about is Toxoplasma gondii. The cat is the definitive host. This is an intestinal parasite acquired through eating small animals, so the cat has usually eaten a mouse or a mole or something like that.
The oocyst of the parasite is shed through the feces of the cat. The methods in which humans usually come in contact with this parasite is through ingestion of the oocyst through infected food, or through hand-to-mouth transmission from contaminated soil, or even cat litter. Also, humans can come in contact with this parasite through undercooked infected meat, because sheep, pigs, and rabbits can get the parasite. Another way that humans can come in contact with it is through congenital transmission. This is transmission from mother to baby. If mother is newly infected during or just before pregnancy, this can cause things like miscarriage, stillborns, or congenital toxoplasmosis. And the toxoplasmosis in the babies can cause or affect the size of the baby's head, so it can cause enlargement or smallness of the head. As far as the life cycle for Toxoplasma in humans, it's asexual reproduction only. The oocyst, you can see what they look like that would be passed in the fecal right here. There's a stage called tachyozoites that develop and divide in the tissue and release, and they're able to be released and invade new cells. And here's examples of the tachyozoites right here in the top right hand corner. And then there is a cyst stage containing bradiozoites that form in the brain and other tissues. And here is a cyst containing those bradiozoites in this lower right hand corner. Here's the life cycle of Toxoplasma, but before we get into that, let's look over here. Toxoplasma lab ID, you can do serological testing, but you can also do tissue biopsies and see evidence of Toxoplasma gondii. And this is showing a cross-section of the central nervous system. So this is a tissue biopsy showing Toxoplasma gondii. You can see the little cyst in the tissue. As far as the Toxoplasma life cycle, of course, the cat is the definitive host, so the humans do not have to be involved at all. So the life cycle would be the cat eating a small mammal, bird, and the cyst would be in the muscle tissue. But occasionally humans come in contact with it, maybe clean the litter box, contaminated food or water, and they end up in the human host. Or another route would be, of course, eating contaminated or infected undercooked meat from a pig, sheep, whatever that has Toxoplasma gondii. And it's also showing a route might be from contaminated blood, or you can get it through that transmission of mother to baby that we talked about a while ago. This slide is a general overview of toxoplasmosis. So you have the Sporozoa, which is the spore-forming part that invades the intestinal cells. Then you have the tachyozoites that form. This happens in the tissue, and they reproduce asexually, the parasite does. And remember, cells can rupture, and everything spreads throughout the blood, and it can cross the placental barrier to the baby, so it affects the fetus. And other cells can be penetrated, such as the brain and other tissues, because the cyst contains those bradiozoites, and it can affect other organs. In immunocompromised patients, the effects can even be more severe. Patients with central nervous system disease can have retinochoroiditis, which is an infection of the eye, or pneumonitis, which is an inflammation of the lung tissue. And also you can have the congenital toxoplasmosis that we talked about. Most cases are subclinical, meaning they may not have signs or symptoms that are detected by a physical exam at birth. So at the end of the day, just be careful cleaning out those litter boxes, especially if you're pregnant. The next sporozoan in the subclass Coccidia is Sarcocyst. Humans, dogs, cats can become definitive hosts if they eat undercooked or raw meat. The host is usually beef or pork containing sarcocyst. Okay. Humans can be accidental intermediate host by ingesting the oocyst, and the parasite can go on to form sarcocyst in the muscle tissue. The oval oocyst of the parasite contain 2 sporocysts. So these sporocysts are basically the same thing as the sarcocyst. The sarcocyst is just more of a specific term for sarcocystis. Remember, all of our parasites in the sporozoan category are spore-forming. And the 2 sporocysts within sarcocystis contain 4 mature sporozoites. The sporocyst can measure anywhere between 9 to 16 microns to 8 to 12 microns. They're kind of elongated or oval in shape, and they can be seen free in feces. And that's what you're looking at over here. This is an example of sarcocystis in feces. And you can see there's a lot of bacteria here, so that we know this is a fecal sample. Here's a diagram showing the life cycle of sarcocystis. Again, humans are not the definitive host. The definitive host would be things like cattle or pigs. Humans become infected when we eat undercooked meat, and the parasite is within the meat or muscle tissue that we consume. So a lot of the life cycle is going to be down in the gut of the human, and we're going to pass the sarcocyst or the sporocyst in our feces for the life cycle to continue. The next in the subclass Coccidia is Cystoisospora belli. It was formerly just known as Isospora. This one is pathogenic to humans. Humans are the only definitive host. You have both forms of asexual reproduction, and you have spore forming in this parasite. So you do have schizogamy, which is our asexual reproduction, and sporogamy, the spore formation. The transmission is usually through the sporulated oocyst in fecally contaminated food and water. Human infections can cause anything from anorexia, nausea, abdominal pain, diarrhea, and possible malabsorption. For the labidae, you usually have microscopic demonstration of large typical shaped oocyst, and you can see what that looks like over here in the top right hand corner. Also, you can see what the sporocyst looks like down here, and you can see the 2 spores in Cystoisospora down here in the bottom right hand corner. The next 2 that are found in the subclass Coccidia are Cryptosporidium parvum and Cryptosporidium hominis. Cryptosporidium parvum is found in animals and humans, but hominis is only found in humans. These usually invade the gastrointestinal mucosa of the vertebrate host. These usually have 8 myriazoites within the schizonots. The oocysts are auto-reinfective. So what does that mean? It can self-reinfect. So reinfection of the organism that has already passed through the infected state can keep on happening, so it can repeat within the host. Oocysts in the feces has 4 sporozoites. So the oocysts of the species are indistinguishable. So in the feces, you're not going to be able to tell parvum from hominis as far as the oocyst. You can use Sheather's sugar flotation for recovery of these eggs for methodology. The eggs are going to be lighter than the solution, so the eggs are going to float up to the top of the solution where you're going to be able to collect them and see them under the microscope. You can use the stain modified acid fast and the oocysts are going to stain positive, and when they stain positive, that's probably going to be a reddish or a pinkish color. If you have any confusion with these parasites with yeast, you can use iodine to help differentiate. Parasite eggs should not stain with iodine. Okay, for Cryptosporidium parvum, clinical features: humans are the only definitive host, but they can be found in other animals as accidental hosts. Most cases are asymptomatic, but you can have fever, diarrhea, and abdominal pain in patients. For the lab ID, you have a microscopic demonstration of the large shaped oocyst, and that's usually the basis for diagnosis. You see the oocyst in your feces or fecal samples. Like I said before, you can use modified acid-fast stain to help ID Cryptosporidium parvum, and your Cryptosporidium is going to stain positive, so they're going to be that reddish, pinkish color against a blue-green black background. So those oocysts are these red, pinkish dots you see in the center here, and all this bluish color is background. Okay, here is our last parasite in the subclass Coccidia for the sporozoans. It's Cyclospora. The clinical features is it's self-limiting, so the diarrhea is only going to last a few days, but relapses often occur. Transmission is usually indirect through contaminated food or water. A lot of times people end up getting this through berries, such as raspberries or strawberries. Lab IDs include wet mounts. Also, you can use that modified acid-fast stain that we've talked about to see the oocyst in stool samples. It must be measured carefully to distinguish from your Cryptosporidium. Your Cryptosporidium is only going to measure about 4 to 5 microns in range. Your Cyclosporia is going to be in the range of 8 to 10 microns. So your Cyclosporia is going to be larger. Here is the life cycle, or the diagram for the life cycle of Cyclosporia. Again, we get the oocysts from eating some contaminated food or water. Again, a lot of times it's from berries that we've consumed. And the oocysts are going to pass through the gut and through the feces. That feces can recontaminate, a food source like berries, and the cycle can happen all over again. So this slide is just showing a good comparison of some of our Coccidia species using different forms or methodologies for ID. This is our Cryptosporidium. It would be our smallest. Here's our little scale, our 10 micron scale. Here is Cyclosporia, and then down here is Isosporia, or Cystoisosporia is what it's called now. Take note that images A, C, and F are stained with the acid-fast stain. B, D, and G are just bright field microscope images of the parasites. And then images E and H are using UV fluorescence to look at the parasites. Alright, in sporozoans, let's move on to the category of Microsporidia. These are tissue parasites that usually affect immunocompromised patients. These are obligate intracellular parasites, meaning they need a host to live. They can inhabit all body tissues. They consist of 7 named genera, and so basically this is a catch-all genus. Some infections may be acquired through the ingestion of spores. Microsporidia have been found in the central nervous system, eyes, lungs, kidney, myocardium, liver, the adrenal cortex, and some have been associated with other diseases such as leprosy, tuberculosis, schistosomiasis, which is caused by a parasitic flatworm, malaria, and even Chagas disease. In immunocompetent patients or normal patients, this parasite is usually self-limiting, meaning that the diarrhea only lasts a few days. The next sporozoan that we're going to talk about is Pneumocystis jirovecii, and it was once thought to be a sporozoan, but now it's really classified as a fungus. This is a opportunistic infection of immunosuppressed patients, and these are usually patients with AIDS a lot of times. It can cause interstitial plasma cell pneumonia, or PCP. It causes non-productive cough and shortness of breath in patients. For the lab ID, we usually use specimens of a lung biopsy or induced sputum. The stains that can be used is Gamori silver, and the cyst walls are going to be black, and they're going to have a punched out ping pong ball appearance, and that's what you're seeing over here. Those outside walls are black, and then they might have a little divot like a punched out ping pong ball. And then the second stain that you can use is the Giemsa stain, and the cyst itself for the outside does not stain, but you can have intracystic bodies for the insides that stain a pinkish color. As far as the life cycle for Pneumocystis, you can have a troph, and you can have the cyst forms, and the cyst is the infective stage, and it usually enters through a respiratory route. And you can see this is Pneumocystis in a lung tissue sample, and you can see the parasite looks like a little ball, and it has like a little divot or a punched out area that looks like a squished or a punched ping pong ball. This will conclude our lecture on sporozoans, and I will see you over in the next lecture. If you have any questions or need clarification on anything, don't hesitate to reach out.